माइक की प्रॉपर्टीज में गया ना मैं। ओके, डिवाइस कौन सा करूँ? माइक्रोफोन, वेबकैम या कौन सा माइक्रोफोन?
Yeah, hello. I haven't stopped streaming while, yeah. So there was some problem with the mic. Anyway, so yeah, so like I said, we are discussing the transport of gases. Okay, so we'll first start with the transport of oxygen from the lungs into the RBC. Okay, how will that happen, and how these RBCs actually transport the oxygen to the various tissues, and then once the RBC delivers the oxygen, okay, the need or role of oxygen in the cells. Okay, what happens to this oxygen in the cells? Okay, then as a result of all the metabolic activity that happens in the cells in the presence of oxygen, carbon dioxide is released, and then we'll look at transport of carbon dioxide to the lungs. Okay, and then finally expiration of carbon dioxide from the alveoli of lungs. Okay, how carbon dioxide is finally released out, and then we'll discuss hemoglobin oxygen dissociation curve. Okay, oxygen hemoglobin dissociation curve. Okay, and towards the end I'll tell you I'll explain Bohr effect and Haldane effect. Okay, so these are the various aspects that we'll discuss today. We'll start off with how the air that we breathe in, okay, containing a mixture of gases. In that mixture of gases you have oxygen. How that oxygen in the mixture of gases is actually, getting into the RBC. We all know that the hemoglobin in the RBC is the one which actually transports the oxygen to the various cells in the body. Okay, first we'll look at that part. Okay, now before actually looking at that aspect, I want to give you a brief explanation about partial pressure of gases. Okay, partial pressure of gases. So if you take a mixture of gases, this mixture will have some pressure, right? Some pressure. It will have some pressure. Now, this pressure which is generated, or this pressure that is present in a mixture of gases, is because of some of the partial pressures of individual gases. That means if I say air is a mixture of nitrogen, oxygen, carbon dioxide and all, the pressure, the atmospheric pressure, the pressure which is created by air is because of the pressure created by nitrogen, the pressure created by oxygen, the pressure created by carbon dioxide. So it's the partial pressures. So the pressure in the total pressure, the pressure created by individual gases is your partial pressure. Okay, now let's look at the partial pressures. Now these values are important. Okay, these values are important. Let's look at the partial pressures that we actually find in the various regions in the body. Okay, partial pressures of oxygen and carbon dioxide. So if we take oxygen, for instance, let's look at the partial pressure of oxygen. And partial pressure is simply represented by PO2. Okay, this is partial pressure of oxygen. Now, if you look at the partial pressure of oxygen in the atmospheric air, okay, in the air, okay, that we are taking in, it is 159 mm of Hg. Okay, and by the time it reaches alveoli, okay, the pressure is 104 mm of Hg. Okay, and in the blood, okay, in the blood, so when the blood is deoxygenated, when the blood is deoxygenated, that is without any oxygen, that is if the RBCs don't have any bound oxygen, okay, and the blood is devoid of oxygen, then the pressure is 40 mm of Hg. And once the blood gets oxygenated, that is blood with the oxygen added to it, the partial pressure of oxygen in the blood which is oxygenated is 95 mm of Hg. And finally in the tissues, it is 40 mm of Hg. Okay, in the tissues it is 40 mm of Hg. Okay, as you can see, the partial pressure of oxygen in the outside air is high. When it reaches alveoli also, it is going down a bit, but it's still high. Okay, from the alveoli it has to move to the blood. Okay, specifically to the RBC of the blood, Okay, so always the gas will move from, right, high pressure or high partial pressure to low partial pressure. Okay, so any gas, it moves from high partial pressure to low partial pressure. So since in the alveoli the partial pressure of oxygen is 104 mm of Hg, it moves from alveoli to the blood, which deoxygenated blood, which has only 90 mm of Hg. Okay, this movement is possible, this diffusion is possible because in the alveoli you have high partial pressure of oxygen when compared to the blood, specifically the RBC of the blood. Okay, now we'll look at the actual events. Okay, now if you look at the actual events, so we all know that the end of the bronchioles, you have many bulging structures like this, which are your air sacs, and these air sacs are nothing but your alveoli. This is your bronchiole, terminal bronchiole. Okay, that is the last branch of your alveoli. Obviously is the last resort of your lungs. Okay, the last component of your lungs. Just before that you have your bronchioles, terminal bronchioles. Okay, the terminal bronchioles will end in many sac-like structures which we call the alveoli. Now this alveoli is actually made up of simple squamous epithelium. This is made up of simple squamous epithelium. So they have a very thin layer of epithelium, right? And this thin layer of epithelium is again covered by another thin layer called the basement membrane. And right next to the basement membrane, we have our blood capillaries. The blood capillaries are right next to the, so the only thing that is separating the air which is actually being collected here, so we have air here, air is getting collected in the sacs, okay, and this air has to move all the way to the blood capillaries over here. So the only thing that is separating here is two layers, that is the epithelium layer and the basement membrane. If it crosses both, then it will simply enter the capillaries. Okay, so this squamous epithelial layer and the basement membrane together, okay, this is called the respiratory membrane. This component is called the respiratory. These two components together, it's called the respiratory membrane. Okay, the alveoli, squamous epithelium and the basement membrane together, this component is called the respiratory membrane. So when the pressure of oxygen, that's the partial pressure of oxygen, is high, okay, in the alveoli, the oxygen will simply diffuse through these respiratory membrane directly into the capillaries. Okay, now what do we have in these capillaries? We have blood in these capillaries. Now what is blood? Blood is, okay, 45% of blood is the formed elements and 55% is plasma. Okay, we have blood here, 55% is plasma, which is simple liquid with many proteins and all. Okay, so almost 95% of plasma is simple water, nothing else. Okay, so the oxygen from here is directly diffusing into plasma first. It's actually entering the plasma. Okay, now very small portion, okay, very negligible amount of oxygen gets dissolved in plasma. So negligible amount dissolves in plasma. Very negligible amount dissolves in plasma. Now what is happening to the maximum or almost all of the oxygen? All of the oxygen from the plasma gets diffused into the RBC. Okay, 45% formed elements will have all the cells. You have your WBCs, RBC and platelets. Okay, so this oxygen which is actually moving from the alveoli into the capillaries, okay, very little portion of it will get mixed in the plasma. Most of it will directly diffuse into the RBC, will directly enter the RBC. Now, what do we have in RBC? Okay, what do we have in RBC? In the RBC, okay, in the RBC, we have hemoglobin. Right? We have hemoglobin. Now, this hemoglobin basically have two components. One is the heme part and the other one is called the globin part. Okay, now the globin part is actually made up of 4 polypeptide chains. 4 polypeptide chains, 2 alpha and 2 beta, and these 4 polypeptide chains, okay, are actually made up of close to 574 amino acids. So altogether it is actually made up of 574 amino acids. Okay, a total of 574 amino acids. Now, if you look at the heme part, okay, each of these 4 globulin chains has one heme part. So we have 4 globulins, and each globulin, 4 globulins and 4 globulin chains. Okay, 4 globulin chains, right? 4 globulin chains. Now each globulin chain, okay, has one heme molecule. Each globulin chain has one heme molecule. Let me just show that. Okay, so you have the RBC is completely filled with hemoglobin molecules, and hemoglobin basically has 4 chains, 1, 2, 3, 4 globulin chains, and each of these globulin chains, 2 beta chains and 2 alpha chains, and each of these globulin chains has one heme molecule. You have one heme, 2, 3, 4 heme molecules, and each of these heme molecules have one iron atom. At the middle, heme is a porphyrin ring, and in the center of this heme molecule you have a single iron atom. Okay, you have one iron atom in each of these heme molecules, and each iron atom and each of these iron atoms can hold a single oxygen atom. So each of these iron atoms can hold single oxygen atom. So the total binding capacity of a single hemoglobin molecule is 4 oxygens. A single hemoglobin molecule can actually bind 4 oxygen molecules. Okay, and we have many such hemoglobin molecules in a single RBC. So this RBC, you know how important hemoglobin is for RBC. It will simply get rid of its nucleus. Okay, it will throw away its nucleus just to accommodate more hemoglobin. So RBCs are the cells which do not have any nucleus. So it simply pushes its nucleus away and fills the entire space with hemoglobin molecules. And each of these hemoglobin molecules can hold 4 oxygen molecules at a time. Okay, can hold 4 oxygen molecules at a time. Right. So you can see here this is what is happening. Okay, for now we'll just look at the oxygen part. So you have the alveolar epithelium. This is the alveolar epithelial layer, right? And then this part is your basement membrane, and right next to the basement membrane or touching the basement membrane is your capillary. The oxygen, when the partial pressure here, like I said, in the alveoli the partial pressure is 104. Partial pressure of oxygen is 104. Since the alveolar partial pressure is high and the partial pressure here, okay, in the deoxygenated blood, the partial pressure is only 40. So the oxygen will simply diffuse from the alveoli through the respiratory membrane, into the plasma. And once it enters the plasma, very small portion of it gets diffused in the plasma. Almost 99% we can say is entering the RBC. 99% is entering the RBC. Okay, now what is happening to this oxygen which is entering the RBC? In the RBC we have hemoglobin, and this hemoglobin, each hemoglobin molecule has 4 iron atoms, and each of these iron atoms will bind one oxygen molecule. Each of these iron atoms will bind one oxygen molecule. So the oxygen which is actually coming from the alveoli, is entering the plasma, from the plasma diffusing into the RBC. In the RBC it is binding to the iron atoms of the hemoglobin molecule. Okay, each of the iron atoms will bind one oxygen molecule. Thus a hemoglobin can actually bind a total of 4 oxygen molecules, and many such hemoglobins are present in a single RBC. Okay, so this is what is happening. This is your alveoli, right? This is your alveoli. Oxygen is diffusing, it's getting dissolved in the plasma, very small portion, less than 2%, as it says here. 98% is actually getting into the RBC. In the RBC it is binding to the hemoglobin and it forms the oxyhemoglobin. Okay, oxyhemoglobin. Now how much? 98% of the oxygen that is coming from the alveoli is forming oxyhemoglobin. Okay, now this oxyhemoglobin, so these RBCs through the blood flow are transported to the cells, to the various cells. Okay, it is transported to the various cells. Now what happens there? Once it reaches the cells, we know that the partial pressure of the cells or the tissue is less. Okay, so if you remember the values that I put up in the beginning, the partial oxygen pressure here is 40 mm of Hg, and in the RBC, once it gets deoxygenated, it is 90 mm. Okay, so the partial pressure of oxygen in the cells is low, because of which oxygen will start first diffusing to the plasma, and from the plasma into the cells. Okay, and if you remember the membrane transport topic that we discussed, I told you clearly that gases like oxygen or carbon dioxide, they don't need the permission of the semi-permeable membrane. They can simply diffuse through the membranes, provided the pressure on either side of the membrane is on one side is greater and the other side is low. Okay, when you have such a situation, the gases like oxygen, carbon dioxide, nitrogen can simply diffuse from one side of the membrane to the other. So the gas from plasma, the oxygen gas from plasma will simply diffuse into the cells. Okay, now we have oxygen in the cells. So we started with alveoli, and from the alveoli the oxygen entered the plasma, and from the plasma it entered the RBC. In the RBC it is actually binding to the hemoglobin molecules, specifically the iron molecules of the hemoglobin, and then transported to the cells, and in the cells where the partial pressure is low, it is getting diffused into the plasma, and from the plasma entering the cells directly by passive diffusion, because inside the cells the partial pressure is low and outside the cells the partial pressure is high, because of which it will simply enter the cells by passive diffusion. No energy is required here. Now once the oxygen is inside the cells, what is it doing? Okay, what is the role of oxygen inside the cells? Why do we need oxygen? Okay, why do the cells actually need oxygen? Okay, we need oxygen because all the metabolic processes okay, will yield energy. Okay, the whole purpose of the cell having so many pathways is for yielding energy. Now, in order to get the energy, so it's like this. You take a piece of paper, okay, and then burn it. When you burn it, you are actually getting energy. Okay, now what you have to understand here is the burning process requires oxygen. Okay, it's nothing but oxidation. Burning is nothing but oxidation. Burning process requires oxygen. Without oxygen, fire energy in this case can be light or heat. Okay, which is generated as a result of paper burning. Now the oxygen here is critical because burning is nothing but oxidation. Okay, oxidation of a material. So oxygen is critical. So in order to generate energy in a cell, in order to get energy from any material, it can be from carbohydrates, it can be from proteins, it can be from fats. Whatever is the food material, okay, burning is nothing but oxidation. Oxidation requires oxygen, and this oxygen is taken in. This oxygen is coming from your breathing process. Okay, this oxygen is used, and they are finally burned to release carbon dioxide. Okay, to release carbon dioxide. So this is what is happening here. Okay, so if you take this is in your mitochondria. Okay, let me just tell you what happens before the mitochondria. This is actually a picture of mitochondria. So this mitochondria is actually in a cell. This entire thing is in a cell. Okay, so you have your glucose coming in with the help of transporters, of course. So you have your glucose in the cytoplasm. What will happen to the glucose? The glucose in the cytoplasm will go through glycolysis. Okay, this entire thing is your cell. Will go through glycolysis, and at the end of glycolysis, you will get your pyruvate. Okay, at the end of glycolysis, glucose, which is a 6-carbon molecule, is getting converted into pyruvate, which is 3-carbon molecule. So you are getting 2 molecules of pyruvate. Now what is happening to this pyruvate? This pyruvate is being transported to the mitochondria. This is your mitochondria. Okay, this pyruvate is transported to the mitochondria, and inside the mitochondria, the pyruvate is getting converted into acetyl-CoA. It is going through the citric acid cycle or the TCA cycle or the Krebs cycle, whatever you may call it, and in the process, carbon dioxide is released. Okay, the carbon dioxide is released, and the carbon dioxide which is released out will simply get out of the cell. Like I said, diffusion, simple diffusion. When the concentration of carbon dioxide on one side of the membrane, this is your plasma membrane. On one side of the membrane, the carbon concentration is high, and the other side it is low. Carbon dioxide will simply diffuse from here to outside. So you have your plasma here. Okay, so the extracellular fluid or the plasma. So basically, the carbon dioxide is being generated as a. Now, if you're talking about proteins or fatty acids, ultimately even the fatty acids are directly transported to the mitochondria, because fatty acids are degradation products of your fats. The fatty acids are then converted into acetyl-CoA. Ultimately, carbon dioxide is released. Okay, and the oxygen that we are breathing in is being used for the ultimate process, which is your electron transport chain, releasing the energy. So this oxygen is being converted into water molecule. Okay, so the energy is being released because of that electron transport chain. Because of the electron transport chain, we are getting your energy. And in the entire process, we see lots and lots of carbon dioxide. Even if you take proteins are first converted into amino acids by the digestion process. These amino acids are then broken down to carbon backbones. Okay, the amino group is removed, and the carbon backbone will then again get converted into acetyl-CoA. Okay, the carbon backbone, whatever is remaining after removing the amino acids, right? After removing the amino group, whatever carbon backbone is there, whatever molecule is left, that molecule is converted into acetyl-CoA or any of the citric acid cycle molecules, and finally they are converted into carbon dioxide and released out. So whatever is the process, oxygen is getting into the cells, and inside the cells, because of the metabolic process to release energy, carbon dioxide is being released out. Whether it is carbohydrate, whether it is fatty acids, whether it is proteins, everything is getting converted into carbon dioxide. And for releasing the energy, we need oxygen. The oxygen is simply diffusing from the outside into the cells, and from inside into the cells into the mitochondria, where it is participating in the electron transport chain and finally getting converted into water molecule. And the carbon dioxide is released as a result of the various cycles. Now this carbon dioxide is now sitting in the plasma. The carbon dioxide is sitting in the plasma. So the next aspect that we'll see is how is this carbon dioxide from tissues to lungs. Okay, how is this carbon dioxide being transported? So let me just show you once again. Whether it is carbohydrates, proteins, carbohydrates or lipids, whatever it is, they're broken down to simpler molecules, amino acids, sugars, glycerol and fatty acids, and all of them have acetyl-CoA as an intermediate. All of them will converge at this point, acetyl-CoA as an intermediate. This acetyl-CoA will then enter the Krebs cycle or citric acid cycle or the TCA cycle, and for this process to occur, for the acetyl-CoA to enter the TCA cycle, we need oxygen. Without the oxygen, the acetyl-CoA will not go through the citric acid cycle. Okay, and oxygen is getting in and the carbon dioxide is being released out. This is the carbon dioxide which is being generated in the various cells. Whether you take proteins, carbohydrates or lipids, ultimate thing is your carbon dioxide. And this carbon dioxide is then, where is it going? It's entering from the cells. This is generated in the cells. From the cells, it is entering the plasma. By simple diffusion. By simple diffusion from the cells, from the various cells of the tissues, okay, it will simply enter the plasma. So now we have the carbon dioxide in the plasma. Now, how is this carbon dioxide transported? Okay, now the carbon dioxide is transported by 3 different ways. Okay, unlike oxygen, which majorly gets transported with the help of hemoglobin, which is present in the RBC. Okay, carbon dioxide is not transported by one particular method or one particular means. It is transported by 3 different ways. Okay, now what are the 3 different ways by which carbon dioxide gets transported? So the first is, so whatever all the carbon dioxide is being released into the plasma. Now we have the carbon dioxide, 100% of the carbon dioxide in the plasma. Okay, 100% of the carbon dioxide in the plasma. Now in this 100% of the carbon dioxide in the plasma, right, 7% remains dissolved in plasma. Okay, 7% remains dissolved in plasma and it is transported to the lungs directly by the plasma. Since plasma is actually circulating along with your blood, plasma is very much a part of your blood. So 7% of the carbon dioxide is directly transported along with the plasma. Now what is happening to the 93 other percent? From the plasma, 93% will diffuse into RBC. 93% will diffuse into RBC. Okay, now in this 93%, once it diffuses into the RBC, okay, many events occur. Okay, the many events occur inside the RBC. Okay, inside the RBC many events occur. Now what are the events? We're talking about what events after CO2 enters RBC. Okay, once the CO2 enters RBC, what are the various events that occur? Okay, now the first thing that happens is in the RBC we have an enzyme called carbonic anhydrase. Okay, and RBC has abundant amount of these enzyme. Okay, a lot of this enzyme. Now this carbonic anhydrase, the specialty of this enzyme is to combine carbon dioxide with water. Okay, it's to combine carbon dioxide with water. This carbonic anhydrase combines these two to form, okay, to form carbonic acid and protons. Carbonic acid and protons. Okay, that's the specialty of this enzyme. Okay, now how much of it is actually converted into carbonic acid. Remember how much is actually entering? 93% of total CO2 is entering the, in this 93%, that is entering the RBC, 70% is getting converted into carbonic acid by the enzyme carbonic anhydrase. 70% is getting converted into carbonic acid. Now, what is happening to the rest of 23%? Okay, what is happening to the rest of the 23%? Now, these protons which are released, they bind to the hemoglobin. Okay, so we are talking about what is happening to the 23% of CO2 that is remaining. 70% is getting converted into carbonic acid. These are the events which are actually occurring in the RBC. 70% is getting converted into carbonic acid, and when it is getting converted into carbonic acid, protons are being released. Okay, carbon dioxide plus water in the presence of carbonic anhydrase, you see carbonic acid being formed and protons. And where is this carbonic anhydrase present? It's present in the RBC. It's abundantly present in the RBC. Now these protons which are released will bind to the hemoglobin. And this binding of hemoglobin, this binding of protons to the hemoglobin will cause a conformational change. Okay, it changes the shape of the hemoglobin a bit. Now, as a result of this change, okay, the affinity of the hemoglobin for CO2 increases. The affinity of the hemoglobin for CO2 increases. So this 23% which is there, it is actually binding to the hemoglobin, and that 23 which is actually binding to the hemoglobin is forming the carboxy hemoglobin. Okay, this forms the so this basically 70% is getting converted into carbonic acid, 23% is binding to the hemoglobin, Now what is happening to the 70%? This 70% carbonic acid, okay, this 70% carbonic acid, what happens here is, this carbonic acid is then exchanged with, okay, so we have in the RBC we have the carbonic acid, okay. Now this is the HCO3 is exchanged with the chloride ions. Okay, the HCO3 is exchanged with the chloride ions. So carbonic acid is going out and the chloride ions are coming in. Exchange will happen. Okay, so here you have HCO3 minus coming out and then chloride ion going in. The bicarbonate ion coming out and the chloride ion going in. And in the process, it is actually releasing protons. So basically you are getting your bicarbonate out and the chloride ion is actually going in. So this is called, this is in the RBC. The carbonic acid which is formed in the RBC is being released out as bicarbonate, okay, and in the process it is chloride ion is actually getting in. So exchange reaction will happen. So this is called the chloride shift. This is called the chloride shift and is also called as hamburger phenomenon. This is called the hamburger phenomenon. Okay, so the chloride shift will happen and ultimately the bicarbonate is actually released into the blood. Okay, the bicarbonate is released into the blood. That is, now this 70% is in the form, this is 70% in the plasma. So in the plasma, as of now we have 7% dissolved carbon dioxide, 7% dissolved carbon dioxide, 70% in the form of bicarbonate ions. 70% in the form of bicarbonate. Where is this coming from? This, the carbon dioxide, the 93% carbon dioxide which entered the RBC got converted into carbonic acid and then finally released again from the RBC into the plasma as bicarbonate ions. Okay, so this is coming from your RBC. This is coming out from the RBC. Okay, now in the RBC, the remaining 23% is in the RBC bound to hemoglobin. It's bound to the hemoglobin. So this is what is happening inside the RBC. Now, how is the removal of carbon dioxide at lungs? Removal of carbon dioxide at lungs. What will happen? So, if you remember, the partial pressure of carbon dioxide, okay, is like this. Okay, I haven't given you the numbers. In the air, okay, we're talking about PCO2, partial pressure of carbon dioxide. In the air, it is 0.3. Okay, the minute it reaches the alveoli, it is 40 mm of Hg. Okay, partial pressure of carbon dioxide in the alveoli is 40 mm of Hg. Then in the blood, which is deoxygenated, in the deoxygenated blood, okay, it is 45 mm of Hg. And in the blood which is oxygenated, it is 40 mm of Hg. And in the tissues, it is 45. So since it is high in the tissues, it is able to diffuse into the blood plasma. Okay, now in the blood plasma, okay, you have two different categories, in the deoxygenated blood and the oxygenated blood. And finally in the alveoli, it is still 40 mm of Hg. Okay, now in the deoxygenated blood, it is higher. In the deoxygenated blood, when there is no oxygen bound to the hemoglobin, okay, it is, so in other words, when the RBC binds oxygen and delivers oxygen to tissues, it becomes deoxygenated. And in the deoxygenated, the partial pressure of carbon dioxide is 45 mm. Okay, so this is the 45 mm. So this deoxygenated RBC will now carry the carbon dioxide and will come to the lungs. And in the lungs, it is 40 mm of Hg. In the lungs, it is 40 mm of Hg. So the partial pressure here is low. When compared to the deoxygenated blood, the partial pressure is low. Okay, so let's see all the events that occurred so far. So because of the various cellular processes, various metabolic process, oxidation of carbohydrates, lipids, or proteins, carbon dioxide is being released. That carbon dioxide will enter the plasma by diffusion. 7% of it remains dissolved in the plasma. Okay, remaining 93% will enter the RBC. This is your RBC. Once it enters the RBC, it combines with water to form, in the presence of carbonic anhydrase, to form carbonic acid. This carbonic acid then splits into bicarbonate and protons. Okay, carbonic acid splits into bicarbonate and protons. Now this proton will actually bind to the hemoglobin, right? This proton will bind to the hemoglobin. Binding of proton to the hemoglobin causes conformational change in the hemoglobin, which now binds carbon dioxide. Okay, and this becomes carboxyhemoglobin, right? Carboxyhemoglobin, right? Now what is happening to the bicarbonate ion? The bicarbonate ion is being exchanged with the chloride ion. So the RBCs are simply pushing out the bicarbonate ion and taking in the chloride ion. Okay, and this part is what we call the chloride shift. So ultimately in the plasma, we are getting 70% plus 7%, 70% of carbon dioxide in the form of bicarbonate and 7% of carbon dioxide in the form of dissolved carbon. Now this plasma along with the RBCs, okay, so this 23%, this 7% and this 70%, everything is actually moving in the blood. And this blood is being transported to the lungs. Okay, is being transported to the lungs. So you have bicarbonate in the plasma coming to the lungs. You have dissolved carbon dioxide inside the plasma coming to the lungs. And you have RBC with the carbon dioxide bound to the hemoglobin coming to the lungs. So all the three components are actually finally reaching the lungs. Now what are the events that occur once all these three components reach the lungs? Okay, now the three things that occur here are, now we have 7% of carbon dioxide in the plasma. Right? Now, since the partial pressure of carbon dioxide in alveoli is less than the partial pressure of carbon dioxide in the plasma, okay, so this 7% carbon dioxide will simply diffuse, okay, from plasma into the alveoli. 7% dissolved carbon dioxide simply diffusing into the alveoli. No issue here. The first component is taken care of. So from the alveoli, when we expire, when we release the gas outside, this carbon dioxide will go out. Okay, just because of the difference in the partial pressures, since the partial pressure of plasma is high and the partial pressure of alveoli for carbon dioxide is low, the carbon dioxide, dissolved carbon dioxide, is simply diffusing from the plasma into the alveoli and being released out. Now, what is happening to the bicarbonate? Okay, what is happening to the bicarbonate? What is happening to the bicarbonate? Now, this bicarbonate, okay, the minute the plasma dissolved carbon dioxide goes down, okay, so in the plasma we had dissolved carbon dioxide. Now this is being released into the alveoli. So what will happen? So this is in the plasma. So what will happen? In the plasma, now PCO2 will go down. PCO2 will now go down. Okay, PCO2 will go down. Then what will happen is there will be exchange of, okay, when the PCO2, when the plasma carbon dioxide concentration, partial pressure goes down, okay, because the dissolved carbon dioxide is being released into the alveoli, the plasma partial carbon dioxide concentration will go down. This causes reverse chloride shift. This causes reverse chloride shift. That is, till now what happened in the initial stages, what happened? The chloride ion is being pushed in. Okay, the chloride ion Cl minus is getting in, and the bicarbonate, this is at the tissues. Okay, at the tissue level, what is happening? HCO3 minus is being pushed out and chloride ion is being taken in. So inside the RBC we have chloride ion. Now this is at the lungs. This RBC has traveled and reached the lungs. Now what is happening? The partial pressure of plasma for carbon dioxide is going down. So the chloride ions here are exchanged with the bicarbonate ions which are present outside. That is a reverse of what we saw in the tissues. Okay, the reverse of what happened at the tissues. So at the lungs, what will happen? This is your RBC. This is your RBC. Okay, at the lungs, the bicarbonate ion is taken back inside. Okay, now the bicarbonate ion is coming back inside. So this is what is happening. So when the partial pressure of carbon dioxide is going down in the plasma, the chloride is pushed out of the RBC and bicarbonate ions are taken in. Okay, we have bicarbonate ions inside. Okay, now these bicarbonate ions, what will happen is these bicarbonate ions will then combine with the H plus ions, okay, which are being released by the carboxyhemoglobin. Okay, so this will simply release the protons and that proton will bind to the bicarbonate ion. So it again forms the carbonic acid. This carbonic acid is again converted into, by the same enzyme, is converted into water and carbon dioxide. And the carbon dioxide is released back into the plasma. And from the plasma, it will simply diffuse into the alveoli. This is your alveoli. Okay, now when the proton is being released, when the protons are released from HbCO2, okay, when the protons are released, what will happen? The conformational change of hemoglobin goes back to its original. Okay, the hemoglobin will revert back to its original shape. When it reverts back to its original shape, it can no longer bind the CO2, or its affinity for CO2 will go down. So that CO2, the 23% CO2 which is actually bound to the hemoglobin, is also released. So entire 70% from bicarbonate and the 23% from the carboxyhemoglobin are being released as dissolved carbon dioxide into the plasma, and from the plasma directly diffuses out. Okay, so that is how the carbon dioxide is actually released. So just to give you a quick summary of carbon dioxide. So you have 100% of carbon dioxide entering the plasma, and in the plasma you have 7% dissolved, right? 93% into RBC. In this 93%, 70% is converted to bicarbonate and released into plasma, 23% bound to hemoglobin. 23% remains bound to hemoglobin. Now all these components will move to the lungs, and near the lungs what will happen? In this 70%, this 70% which is now in the plasma will go back to the RBC. So this 70% will move back inside RBC. Then again gets converted into, okay, the 70% bicarbonate will get converted into carbon dioxide plus water. This carbon dioxide is released into the plasma, and from the plasma this is released into the alveoli, diffuses into alveoli. Now what is happening to this 23%? When the protons are removed when the partial pressure of carbon dioxide is low, the protons are actually removed from the hemoglobin. When the protons are removed, this causes a conformational change in the hemoglobin, and the hemoglobin will now split into Hb plus CO2, and this CO2 then enters the plasma, and from plasma to alveoli, and finally released out. So this is the fate of carbon dioxide from the tissues all the way to the lungs, and from the lungs into the alveoli, and finally disposed of. Okay, so that is about the transport of oxygen to the various tissues and the transport of carbon dioxide from the various tissues to the Then finally, a few aspects that we still have to discuss is oxygen, hemoglobin, dissociation curve. Oxygen, hemoglobin, dissociation curve. Now, so a curve which is actually generated with percentage saturation of hemoglobin with O2, of course, against partial pressure of oxygen. Okay, increasing partial pressure of oxygen. So you continuously increase the partial pressure of oxygen and see what happens to the hemoglobin. Okay, whether it is getting fully saturated or not. What do we mean by full saturation? So like I said in the beginning, hemoglobin basically has 4 iron atoms, and when each iron atom can actually hold 4 oxygen molecules. So if all the iron atoms, if 4 iron atoms are bound with 4 oxygen atom, we'll say that this hemoglobin is saturated. Okay, under normal arterial pressure. So if you look at the normal oxygen pressure inside the arteries, okay, which actually carries the oxygen to the various blood, most of the hemoglobin is completely saturated. However, when the oxygen partial pressure is low, okay, only 3 oxygens may be bound to the 4 iron atoms, or only 2 oxygen may be bound to the 4 iron atoms. Okay, so in oxygen molecules, not atoms. So in such situations where the required number of oxygens that is lower than what the hemoglobin can actually carry, we say it's not completely saturated. So when we increase the partial pressure of oxygen continuously, okay, we'll see a graph like this. So in the initial stages, as you increase the concentration of oxygen, more and more hemoglobin will actually bind the oxygen molecules and get saturated. Okay, at some point when the oxygen concentration is high enough that all the molecules are already saturated. Beyond that point, even when you give more and more oxygen, you will actually see a plateau. Okay, so increasing oxygen concentration will not make any difference because all the hemoglobin molecules in the RBC are already saturated at this particular point. Okay, so all of them are saturated, so increasing the oxygen pressure will not. However, in the lower concentrations, gradually the saturation or the number of oxygen molecules that an iron molecule in the hemoglobin can bind will gradually increase by increasing the partial pressure of oxygen. Okay, by increasing the partial pressure of oxygen. Right. So this is the oxygen hemoglobin dissociation curve. Okay, so in the initial stages, when you increase the partial pressure of oxygen, the amount of oxygen bound to the hemoglobin will also increase. After a certain point, when all the hemoglobin molecules inside the RBC are completely saturated, that is completely bound with oxygen molecules, increasing the partial pressure of oxygen will not make the hemoglobin molecules to bind more oxygen. The maximum that they can carry is 4 oxygen molecules. So increasing partial pressure of oxygen beyond a certain point will not increase the amount of oxygen that can actually bind to the hemoglobin. Okay, so this is your oxygen hemoglobin dissociation curve. Now this oxygen hemoglobin dissociation curve, okay, will depend on various factors. Okay, the oxygen hemoglobin dissociation curve will depend on various factors. Various factors actually influence this curve. Okay, the first is partial pressure of carbon dioxide. Partial pressure of carbon dioxide. Then the second one is pH. The third thing is your temperature. Okay, what are we talking about? We're talking about the factors that influence oxygen hemoglobin dissociation curve. Oxygen hemoglobin dissociation curve. Okay, and this you won't find this in most of the textbooks, but there are a couple of other factors which actually influence this. One is 2,3-diphosphoglycerate. This is a chemical compound which is actually found in the RBC. Even this influences the oxygen hemoglobin dissociation curve, that is the binding status, the binding ability of hemoglobin, the oxygen binding ability of the hemoglobin. Okay, and the other thing is unusual hemoglobins. Unusual, when I say unusual hemoglobins, in the fetal stage you have fetal hemoglobin, not the adult hemoglobin, you have fetal hemoglobin. Okay, and in people with thalassemias and sickle cell anemia and all, they have a different hemoglobin. Okay, in those hemoglobins the oxygen binding capacity will be different from a normal hemoglobin. So these factors actually influence the oxygen binding capacity of hemoglobin. Okay, now how do they influence? Okay, what is their effect? If you look at their effect, so you can see here, this is the effect of partial carbon dioxide. Okay, so when you increase the carbon dioxide concentration, so you can see here, this is for 20 mm of Hg. This is for 20 mm of partial carbon dioxide concentration. When you increase the partial pressure of carbon dioxide, okay, this is the normal one. This is normal at normal partial pressure of carbon dioxide, and this is when you have high CO2. When you have high CO2, okay, you can see the saturation of hemoglobin going down. Okay, the saturation of hemoglobin will go down. When you decrease the carbon dioxide concentration, the partial pressure of carbon dioxide, when you decrease the partial pressure of carbon dioxide, in other words carbon dioxide concentration, the oxygen binding ability of the hemoglobin will increase. This is when you decrease the concentration of the carbon dioxide. it is actually increasing. When you increase the concentration of carbon dioxide, it is going down. This is under normal conditions, normal carbon dioxide partial pressures. You will see this graph. So carbon dioxide is one factor which actually influences, okay, whether your hemoglobin binds oxygen well enough or not. Okay, now what are the factors? The other factors that we'll see here. Okay, now let's look at the pH first. What is happening to the pH? This is at normal pH. The purple line in the middle is the normal pH. When you increase the pH, okay, from 7.4 to 7.6, okay, the affinity is also increasing. The affinity of hemoglobin for oxygen is increasing when you increase the pH of the blood. Okay, now when you decrease the pH, the affinity of oxygen, the affinity of hemoglobin for oxygen is decreasing. That means let's say in the lungs, okay, in the lungs, right, in the lungs, let's say the pH is 7.4. The pH is 7.4. This is good. So at this pH, oxygen will simply diffuse and bind to the hemoglobin. No problem. Okay, in the plasma, let's say from the lungs, in the plasma also it is 7.4. So simply diffuse and bind to the hemoglobin of the RBC. However, when it reaches the tissues, let's say in the tissues, the pH, okay, when the RBC finally reaches the tissues, let's say the pH there is 7.2. What will happen when the pH is 7.2? The diffusion, the affinity between hemoglobin and the oxygen will go down, because decreased pH will decrease the affinity. When the pH decreases, the affinity will decrease. So in the tissues, if the pH is 7.2, then oxygen is being released very quickly. Okay, let's say in some other tissue the pH is 7, then even more quickly or even more faster will be the release of oxygen from the hemoglobin. Okay, so in other words, the more acidic the tissue gets, the quicker will be the release of oxygen from the hemoglobin. Okay, now the reason why I'm saying this is, in the tissues, when you don't have enough oxygen, okay, we see the accumulation of lactic acid. Because you're getting energy in a different route, okay, through the fermentation process. Okay, the pyruvate is getting converted into lactic acid. I don't want to get into the details, but just remember this: the muscle cramps and all that you get, okay, when you don't have enough oxygen supply to your tissues, okay, the cramps are because of lack of oxygen supply to the tissue. Now what happens in the lack of oxygen supply to the tissues? In the lack of oxygen supply to the tissues, energy is generated by converting your pyruvate molecules into lactic acid. So lactic acid starts to accumulate in the tissues, and this lactic acid is the one which is actually responsible for the cramps, the pain and all. Okay, so in those tissues those tissues desperately need oxygen. So when the lactic acid starts to accumulate, the pH in the tissues will go down. When the pH go down, okay, more and more oxygen will be released from hemoglobin. So these tissues will actually get more oxygen when compared to other tissues. The tissues where the acidity is more, they need more oxygen. Okay, and since the acidity is more, the pH is going down. Okay, pH is going down. That means the hemoglobin will release more and more oxygen in this region when compared to the other parts of the body. Suppose you are running, so your calf muscles, your leg muscles will be the ones which require maximum oxygen. So what will happen? More and more lactic acid is produced in the legs. So this causes a lowering of pH, and more oxygen is needed because your need for energy is there. Okay, so the lactic acid will lower the tissue pH, so more and more hemoglobin will reach there and release more and more oxygen. So that's how the tissues are ensuring that they get more oxygen by lowering the pH. Okay, so that is the effect of pH on the oxygen hemoglobin dissociation curve. So increase in pH will increase the affinity of oxygen for hemoglobin, and a decrease in pH will lower the affinity of affinity between oxygen and the hemoglobin. Now, what happens to the temperature? What is the effect of temperature? In the case of temperature, when you decrease the temperature, the affinity will increase. Okay, when you decrease the temperature, the affinity, in other words, okay, if you bring down the temperature of your plasma to 20 degrees, okay, or your RBC to 20 degrees, it will bind more oxygen than when it is actually binding at 37 degrees centigrade. Okay, the affinity of oxygen and the hemoglobin will increase when you bring down the temperature. When you increase the temperature, the affinity will go down. It will quickly deliver the oxygen. It won't bind the oxygen that strongly. Okay, decreased affinity, so it will simply deliver the oxygen very faster. So this is the effect of the major three factors. Okay, and then if you look at the final aspect, which is the effect of glyceroldehyde 3-phosphate, 2,3-diphosphoglycerate. Okay. So accumulation, I'll tell you here itself. Yeah, accumulation of DPG will lower the affinity. Accumulation of DPG will lower the. So if you have more DPG inside, the affinity will go down. Okay. So let me see if I have the graph for that. Just give me a second here. In the meantime, just look at this graph. So this is in the case of normal blood, and when the partial carbon dioxide concentration increases, you see the graph, the saturation curve going down. When the carbon dioxide concentration decreases, you see the graph going up. Okay, so just give me 1 second. Okay, as of now I don't have the graph for the effect of DPG on the saturation of hemoglobin, but I can tell you this, accumulation of DPG will lower the oxygen affinity. Okay, accumulation of DPG will lower the oxygen affinity. Okay, so and then I'll tell you the final aspect, which is Bohr effect. Just a couple of minutes, will be done. Okay, now what is Bohr effect? Bohr effect basically is the decrease in blood pH. I told you just now, decrease in this was observed by Christian Bohr. So he was actually studying the effect of pH on the saturation of hemoglobin. Okay, so what he did was he took the blood cells and then put them under different pH, okay, and checked how the hemoglobin is able to bind the oxygen. So what he found was decrease in blood pH, okay, results in a lower affinity of hemoglobin for oxygen. I'm sorry, for oxygen. Okay, so this is what he observed. Decrease in blood pH. When the blood pH decreases, the affinity of hemoglobin for oxygen will go down, and this is called the Bohr effect. Essentially what I showed you in the graph. Okay, so now why is this happening? This is happening because if you remember the initial part, I told you H+ ions bind to hemoglobin, causes conformational change, and this conformational change will cause the carbon dioxide to bind but not oxygen. Okay, conformational change will cause the carbon dioxide to bind but not oxygen. So the affinity for carbon dioxide will increase but not for the oxygen. And when you say when you're saying decrease in pH, okay, decrease in pH is more of H plus ions. Decrease in pH is becoming more acidic. More acidic is more H plus ions. Okay, when you have more H plus ions, those H plus ions will actually bind to the hemoglobin, cause a conformational change, and this conformational change will cause, is the reason why the affinity between the hemoglobin and the oxygen is reduced. Okay, so this is your Bohr effect. Then you have Haldane effect. Haldane effect. Haldane effect is decrease in the affinity of decrease in the affinity of hemoglobin for H plus ions, that is the protons, H plus ions, protons, and carbon dioxide when O2 binds to hemoglobin. Okay, so when you have enough oxygen, oxygen will actually bind to the hemoglobin. Okay, when there is oxygen, the conformation of hemoglobin will not change, so it will not bind to the protons. Okay, or the affinity, when the oxygen is around, the affinity of the hemoglobin for H plus ions and carbon dioxide will be low, and this decreased affinity, okay, is what we call the Haldane effect. Okay, this decreased affinity is what we call the Haldane effect. So that wraps up this session, and tomorrow when we meet, we'll talk about a completely different topic. Yeah, so a few questions that were asked were, why squamous epithelium layer? The reason is wherever you need moisture, okay, because you need moisture there, and that moisture is provided by squamous epithelium. Moreover, that's one reason. Moreover, these squamous epithelial layers have very thin membranes, okay, which allows the diffusion of oxygen from the alveoli into the capillaries very easily. Okay, then why does negligible amount? Because why does negligible amount get dissolved in plasma? Okay, one question that was asked is why does because the affinity, I can say this. So this is about oxygen, right? So the affinity of oxygen towards hemoglobin is much higher than the affinity of oxygen towards the plasma. Okay, that's the reason why only a small percentage gets dissolved in the plasma. Rest of it will enter the RBC and gets attached to the hemoglobin. Okay, so that's the reason why only a negligible amount remains dissolved in the plasma. Why does alveoli always keep residual volume of? Yeah, so alveoli, it's not just the alveoli that keeps the residual volume. Okay, so some of the oxygen will remain in the lungs. It cannot be pushed out. Okay, and see, whatever we do, we cannot expect. Let's look at the pressures here. Okay, if you look at the pressures here, the partial pressure of oxygen, okay, in the alveoli is 104, right? And the partial pressure of oxygen in the blood is 40. So this is in alveoli and this is in plasma or the blood, okay, deoxygenated blood, right? So what will happen here is, you can't expect all of the gases or all of the air to be moved to the next or to be transferred to the plasma, because diffusion will occur till the equilibrium is achieved. So you have 40 here, so you can't expect all the 104 to be transferred to the other side. Okay, so all the oxygen molecules cannot be transferred, because at some point equilibrium is achieved, and once the equilibrium is achieved, some of the oxygen will remain in the oxygen or the alveoli. Okay, because of that reason, okay, you can't expect entire oxygen being transported to the alveoli. Some of the air, some of the oxygen will remain in the lungs or the alveoli as residual gases. Okay, now here also even when the hemoglobin molecules actually go to the tissues, the entire oxygen is not being delivered to the tissues. Some of the oxygen will be retained by the hemoglobin molecules, and this oxygen which is actually retained by the hemoglobin molecules, this is called the reserve oxygen. Reserve oxygen. Okay, so in situations where our breathing blocks or our respiration or breathing process is stopped for a few minutes, we can still survive because there's some residual oxygen or the reserve oxygen in the hemoglobin. So this hemoglobin, this reserve oxygen in the hemoglobin is helping us to survive a few minutes even without breathing. Okay, so that is being supplied to the various tissues and keeping us alive until your CPR can actually kick in. Which transporter does glucose come into mitochondria? Okay, this is somewhat unrelated to the topic, but basically this is what happens. So you don't see the glucose being transported to the mitochondria. The glucose first enters the cells with the help of your glucose transporters, okay, GLUT2. Then in the cells you have cytoplasm. In the cytoplasm, the glucose is getting converted into pyruvate through the glycolysis process, and this pyruvate is the one which is actually being transported to the mitochondria. So you don't see the glucose being directly transported to the mitochondria. It's getting converted into pyruvate in the cytoplasm, and this pyruvate is the one which is actually transported into the mitochondria to finally get converted into carbon dioxide. Okay, so this pyruvate is transported by a complex called pyruvate dehydrogenase complex. It's a big enzyme, and you have a pyruvate transporter along with it. So both of them will actually help in converting this pyruvate into acetyl-CoA, and finally acetyl-CoA is transported into the mitochondria. Okay. So someone asked chloride shift again. Okay, explain chloride shift again. So basically this is what happens. So you have your RBC. The cells are actually releasing carbon dioxide, right? Cells are releasing carbon dioxide. In this carbon dioxide, 73% remains, 7% remains in the plasma, and then 93% entering the RBC. Inside the RBC, what is happening? Inside the RBC, you have carbonic anhydrase, which is converting 70% of it to bicarbonate ions. Okay, and remaining 23% is actually binding to the hemoglobin. Now this 70% bicarbonate, it is actually pushed out of the RBC. This is your RBC. It's pushed out of the RBC. Now it cannot be pushed out directly because this is a negative ion. So you have a transporter here which is actually pushing this out and taking in chloride. Exchange is happening. Okay, so chloride is being taken inside the RBC. At the same time, bicarbonate, another negative ion, is being pushed out. Okay, is being pushed out. So this exchange is happening. So this exchange is what we call the chloride shift. Okay, so the chloride which is there in the plasma is moving inside the cells, inside the RBC, and the bicarbonate is being pushed out. Bicarbonate is being pushed out. This is chloride shift. It is also called as hamburger phenomenon. Okay, so that is all the doubts that you guys had. So hope you understood the session today. So we'll meet tomorrow. Keep following us. Subscribe on our YouTube channel. Follow us on Instagram, Facebook, and of course YouTube. And with that, see you tomorrow.